So I'm going to introduce Amanda and then turn it over to her. So Amanda is the InReach and Outreach Coordinator for the Commons Fund Data Ecosystem Coordination Center, an NIH initiative to increase access and use of the NIH Common Fund data sets. She has quite a broad and eclectic scientific background and has worked previously with communities like population genetics, spectrometry, entomology, genomics, and building tank armor. I love that. She uses these experiences to train scientists to use new computational tools and data types, as well as to build and manage communities. Her interests include statistics, coding, video games, and occasionally tweeting. And I can post that link a bit later. So Amanda, I'll turn it over to you. Thank you.
Yeah. So let's get Zoom going. There we go. So yeah, that was all of my titles. I teach all of the time. So I really like it when people interrupt me whenever you feel like it. I have the chat and the hands and the sound all on, but once I can keep track of everything. So if you see something that you want me to stop on, just yell or type or whatever makes you happy. So I'm going to talk to you real quickly about operationalizing FAIR at the Common Fund data ecosystem. And the Common Fund data ecosystem is this project where our goals are to enhance the ability to ask scientific questions across data sets, to enable uptake, reuse, and addition of Common Fund data and tools, to support the storage, sharing, and sustainability of Common Fund data sets, and to provide training that lets people actually use those data sets. But I always like to start this kind of presentation with backing up and talking about like what even is the Common Fund, because it's not a thing very many people are familiar with. So you probably are aware that the NIH is made up of 27 different institutes and centers, and that each of one of those has its own like deal that it works with. So the National Cancer Institute, they fund stuff about cancer. The Institute of Allergy and Infectious Diseases, they fund stuff about allergies and infectious diseases. The Eye Institute funds eye stuff, et cetera, et cetera. So there's all of these institutes, they all have reasonably narrow focuses. And that's cool, but about 15, maybe even 20 years ago, the ability to do genomic sequencing at a relatively large scale started to really take off. And what happened at NIH is that it became really difficult for a researcher to figure out when they had a new cross-cutting thing that they wanted to do, like they wanted to do genomics on allergies, who to send that funding requests to. And at the NIH, when they got funding requests like this, what mostly happened was the Allergy Institute would say, no, the genomics people should fund this. And the genomics people would say, no, the allergy people should fund this. And so things weren't getting funded. Nobody really knew what to do. So the upshot is that in 2006, Congress created a new IC, and it's called the Common Fund. It's called the Common Fund because they literally took 2%, I think it was, of each of those 27 IC's budgets, pooled it in a common bucket, and said this is going to be what funds things that are these cross-cutting, nobody can agree who should fund it stuff. So in order to be funded by the Common Fund, you're supposed to meet these 5 criteria. It's for transformative, catalytic, synergistic, cross-cutting, unique things, which basically is just a whole bunch of fancy words for saying stuff that fits in more than one IC that everyone was arguing about. So these Common Fund programs are supposed to be developing these innovative, cross-cutting data sets for broad use that wouldn't be funded by any individual IC. So there are, since 2006, there have been about 45 funded Common Fund programs, and Common Fund doesn't fund individual, like one lab science projects. What they fund is large consortia. So the one that almost everyone has heard of is the Human Microbiome Project. So the Human Microbiome Project, they took something like 3,000 people. They took samples from stool. They tried to characterize what the general human condition of intestinal bacterial communities is. They also did some studies later on some disease states, but it was this huge cross-cutting study, and as you can imagine, it needed people with familiarity in genomics, with human health, with microbiomes, with all of these different things. So it's these big cross-cutting things. Another one that a lot of people have heard of is GTEx, and what they do is they are trying to make the baseline RNA level map of every human tissue. So they take very recently deceased cadavers, they take samples from each of 53 tissues, they run RNA-seq analysis on them, and they are trying to build a catalog of what normal RNA-seq levels are for every gene for all of these different tissues. And I'm not going to go through every single one of these, but all of these different programs are these like huge scope trying to answer these cross-cutting questions. Now, there are some issues with the way that these things are funded and that they have evolved over time. So Congress, when they enacted taking a little bit of the budget from each of these places and dumping it in the middle, they said that when you fund something through the Common Fund, you can't fund it for more than 10 years, and that after those 10 years, somebody else needs to pick up the funding. But at no point did anyone figure out who or what that would be. They kind of hoped that an individual IC would pick up the funding on important things, but that actually has run into the same problem as the original funding, which is that all of the different ICs think that a different IC should pick it up. So there's sort of a sustainability issue with common fund data sets. And these data sets were all this transformative, catalytic, synergistic, cross-cutting, unique things. They were all these things that had never been done before, that NIH didn't know how to fund, that needed new data types, that were creating new types of data, creating new file formats, creating all of these new things. And so each of these common fund data sets that has been funded basically got a big chunk of money, and they went and they did their cool research and they made up their own data file formats. They made up their own ways of storing the data. They have all different storage. They have all different ways of describing the metadata. And so the practical upshot is that GTEx, like I said, has made this really cool baseline data set of RNA-seq levels across people. And you can't directly compare it with any of these other Common Fund program data. And you can't actually compare it with your own data very easily, because you would need to reprocess the entirety of the GTEx data in order to make a good comparison. Same thing with HMP. They have this really cool data set, but you can't actually compare it to GTEx. You can't take the two RNA-seq files and look to see what is different between this baseline and this baseline. So we have this problem that because each of these Common Fund programs was created to be these unique snowflake, really cool ideas, that all of them are unique snowflakes that don't talk to one another. So that is why the Common Fund data ecosystem now exists. Our job, my job, is to try to fix this problem, to increase data reuse by essentially making these data sets interoperable and making rules is the wrong word, best practices that as new Common Fund programs are funded, they can use so that interoperability is a thing that happens naturally across data sets going into the future, rather than something we sort of have to backtrack and fill in now. So that's basically what we're doing. We're working on also fixing, trying to deal with that 10-year funding limit and some other things, but the thing I'm mostly going to talk about is this data reuse and interoperability. So real quickly, is this even a useful thing to do? So you could imagine two different ways to sort of jam data together, and I have named them munging and layering because that is how it works in my brain. So munging is the idea of there's some specific RNA-seq data set, there's some other specific RNA-seq data set. They have a whole bunch of things in common, and I just want to put them together and treat them as one really big RNA-seq data set so that I can answer some kind of question, right? I want to take those two data sets and munge them together. And this can be really, really difficult because the data sets either have to have a ton of metadata described so that you can statistically account for the differences between them, or that are not interesting, like blocking effects, or they need to be like basically exactly the same as one another. So munging can be really difficult, but you could imagine other ways to recombine data, which would be something like, I have this really cool, very specific RNA-seq study, and I want to know whether these cells also show differences in some kind of metabolic study. So maybe I want to pull out the serum around these cells and see what the metabolites are there. And maybe I want to also look at the histology of those cells. And maybe I also want to see if in a GWAS these genes pop up in these cells. And so you could imagine taking lots of different kinds of data and sort of stacking them on top of each other in layers to get more information about the thing that you care about. And this is a little bit more of it's a little bit easier of a reach, right? Because all you really need is data that has some kind of overlapping feature. And so with these two in mind, what we did before we started doing really anything at Common Fund is asked whether all of these data sets actually did have things in common that you could, is interop even a possibility here? So this is, and I apologize for this, a very ugly plot because this is super multidimensional data and I just still haven't found a very good way to deal with it. So this is all of those all of those Common Fund programs that I had the logos of. Each one of these is a Common Fund program. And then this is a list of about 80 different tissues. And so if they have a black line at that tissue, they have data on that tissue. And if they don't, then they don't have data on that tissue. So you can see that without reading what every single tissue is, that there is quite a lot of overlap by tissue for different Common Fund programs. So we probably could support something like that layering. We could get different, or sorry, we could probably support something like the munging, right? If there's the same kind of data from links and GTEx and IDG and comp all about the same tissue, maybe we can smush data together that way. And we also looked at other sorts of metadata. So this is sort of where the data came from. There's also quite a lot of overlap between human, right? So all of these tissues came from humans. So that suggests that we could munge things together. There's disease versus healthy. So we have contrasts that we could compare against. And again, we have a lot of this across all of these different programs. And then we asked, well, what if we don't have enough similarity there? What if we just wanted to sort of stack data on top of each other? So this top one is showing different kinds of analyses that happened. So you can see that Links has done analyses with RNA-seq. They've done metabolites, binding, epigenetic, gross imaging, subcellular imaging, and protein structure. And GTEx has whole genome sequencing and RNA-seq and gross imaging, but they have a ton of overlap in tissues. So probably if I went and found the tissue that I care about at GTEx, I could have not just whole genome sequencing and RNA-seq and gross imaging of that, but maybe I could find corresponding data about the metabolites or about the epigenetic information or subcellular interesting imaging or other things like that. So
Amanda,
yeah,
I'm sorry.
go.
No, finish. I was just going to ask a question.
So we thought from the onset that there should be some usable interoperation that we can make out of this data. Florence.
Right. And you may want to answer this later, but watching and listening to this, I wanted to ask you if you think, well, first I wanted to ask you how many PIs are using this or if this is done at a different level. And then I wanted to bring up that one of the projects that we have as the hubs is this COVID Information Commons. And we currently have 990 NSF awards in there. We invite NSF and NIH PIs to present on our monthly COVID Info Commons PI Lightning Talk webinars. And we just got an extension to the award to add NIH awards to it. So we're going to be doing even more between NSF and NIH researchers. And I'm thinking this is just so uber cool. And one of the things where that's part of this new award is to add some new data and metadata search and discovery mechanisms, so that when you put in a keyword search, it doesn't just go into, you know, the metadata for all the awards, the thousands of awards that we'll have in there, but it actually can crawl some data sets too and find some stuff. So I'm thinking your stuff is just so uber cool that it would be cool to maybe have as like a little lightning talk on actually one of our Kick Lightning Talk webinars, if, as long as we're not teasing them and they actually could use it. Like if we say, oh, look at this cool thing, but you can't use it, that wouldn't be a lot of fun. But I'm wondering how PIs would engage with this potentially around COVID.
Yeah, so I'm going to answer that as I go. I'm
Okay.
excited about this.
Oh, good, me too.
So yeah, so we decided, we looked and we thought that this should be like a practical, like there should be something that comes out of this that somebody could actually use. So what we did is we have built a portal that allows you to go and search all of this data using a common model so that you can do data discovery across all of these different data sets. So first I'm going to give you an extremely short tour of the model, just so you can kind of see what we did. And then I'm going to show you a really short demo of the portal and give you the address so that you can use it if you want to. And then I'm going to stop talking. So whirlwind tour of the model. So what we did is year one of this project, we went to each of those programs that I showed you the logo for. We went and physically visited them. We talked to them for a couple of days, asked them everything we could think of about their data and how they store it and what they do to try to figure out what kind of model would work. And then we built a model that everyone can use to describe their data at their portal or at their place. So we don't move the data. We host a metadata search system. And right now we're working on making it go and retrieve those data files and put them somewhere for you. But right now you can search them, and in a couple of months, you'll be able to access them and it'll be great. Oops.
And then where do people save them? Are you providing a repository, like a little sandbox for them, or do they?
We are working on interoperating. We are working on being able to send them to workspaces like Kavatica to work on. But in our portal, you can save your search and all of the information about it to your personal profile.
Very cool. Thank you.
Yeah, so right now we're in the 3/4 or so through year two, where we're building this metadata model post-hosted thing. So this is a little snippet of part of how our model works. We basically picked 3 broad entities, and we've been adding other ones, but we allow people to search by biosample, by subject, or by file. And we have a whole bunch of metadata about each one of those things. So if you are interested in looking for biosamples that are available through the Common Fund, then we basically have a file that the person that has all of this data fills in, and it makes it searchable. And the important thing about this is that we are trying to not make any new standards. So our model is really sort of a monsterization of a whole bunch of standards that already existed. We're just jamming them together into something usable. So what we're really trying to do is make our data more fair, and we use the FAIR principles to sort of build this model out. So we make unique IDs using the IDs that the data already has. We just append a unique bit to the beginning, which is the, usually it's like this would just be GTEX. So we append the name to the front, and then we keep using the identifier they already had so that we get a unique ID. We use persistent IDs, and those can be whatever persistent IDs they already use. So it can be DOIs, min IDs, whatever they already have, we support all of them. And then when we're describing the actual samples, we are using controlled vocabularies that already exist. So for instance, to describe those different tissues, we are using the Uberon controlled vocabulary, which has a gazillion anatomy terms, And so we just pull in Uberon directly, and everyone takes whatever model they normally use to describe their data, maps it as the expert on their data to Uberon. That goes into our portal, and that gives end users the ability to search across all of these data sets using the Uberon terminology, even though that isn't what all of the Common Fund data sets used originally. So we're federating across all of these data sets, but we're not moving any of the data, and we're not forcing everyone to change their internal models. We're just building sort of a map. I said all of this without clicking. Sorry about that. And we did the same thing for all of these different things. So like I said, you can also search by file, same deal, local IDs, names. We can let you search by format, by the data type, by assay type, by MIME type. And the same thing for each one of these, we are using existing ontologies and not making up anything on our own. If you'd like to, go and look at this, I'm going to switch my screen share to here. It is app.nih.cfde.org, putting a link in the chat. And this is our portal. It's completely open to the public. You can see that we have lots of files, biosamples, subjects, anatomy terms, assays, all sorts of things in here right now. And the Common Fund programs that have submitted data as of today are these 9, but we get more all the time. And the way that the portal basically works is if you just kind of want to see what's here without going and doing a deep search, we have a little interactive plot where you can say, like, show me what anatomy terms that you have disease data on, and it gives you, this is by file, so what file counts we have for things about the immune system, reproductive system with prostate, adenocarcinoma, all that sort of thing. So you can do sort of high level, what kind of data exists. If you want to go and find information, if you want to go and actually find those files, you click on the thing that you would like to locate. And we have a faceted search system that lets you search on any of those different things. So if you're looking for, say, expression data, and you want it from the nervous system, you can find out what kind of different files we have. I don't have a preset search path for doing this demo, so I'm just randomly clicking things.
So how would I find data related to COVID? So I went in and I'm grouping by disease.
Yeah, so if we have any, it would be in here. The disease one is the feature to be able to append disease information. We just released like 2 or 3 weeks ago. So I don't think anyone has put any disease information in yet.
Okay.
But there should be some appearing in the next couple of months.
So what I'd like to suggest, Amanda, if you're
Yeah.
comfortable with this, is maybe we could have a separate chat, because I don't want to take over the whole call.
Yeah.
And we could talk about if it would be interesting to work with our COVID Information Commons community, the 990 NSFPIs we have in there right now,
Mhm.
the 800 we'll be adding in the next couple of months, and then the couple of thousand NIHPIs we're going to be adding that have COVID-related research,
Yeah.
and maybe do something with that.
Yeah. No, I would love that.
Okay, cool.
So I'm going to show you guys one quick thing here that's my specific pitch about this talk. So one of the things that we noticed right away when we started doing this is that if you look at the kinds of data that people submitted, you'll find that it doesn't actually look like it's very well harmonized. So, like I said, we specifically used existing ontologies. We specifically gave people like these very specific things that we wanted them to tell us about their data. And we asked the people that are the experts on that data to map it to those ontologies. And you can see, if you spend a little bit of time sort of looking at this, that what happened is that some people just chose data And some people said, oh, well, there's just sequence, even though they could have chosen RNA sequence or DNA sequence or protein sequence. And that we have things like sequence assembly that also probably is just an RNA sequence or a DNA sequence or a protein sequence. And so what sort of happened is that even though we gave people specific ontologies to work with, they all sort of picked and choose within those ontologies in slightly different ways. So what we ended up with was that each program actually has almost entirely non-overlapping data. So this is all from one program, this is all from another program. And this sort of flies in the face of that plot I showed you that said a bunch of this data should overlap. And it's not that it doesn't overlap, it's that because we asked people to pick the best thing that they could in each ontology, and they all chose differently, it basically made the data not harmonized, but in a new way. So what we are doing now is we are implementing a, we are implementing SLIMs. So each of our Common Fund programs now still uses the most specific term, the thing that they think best maps for their ontology, but we have added a second layer of information that you can search by that is perhaps more useful and that sort of brings back that harmonization thing that we wanted to do. So now if I look for things like connective tissue in the anatomy SLIM, it will bring me back all of the things that are under that umbrella term so that users can see what all of the connective tissues were, regardless of what the specific term was that the program chose. And this is really helpful for things like the circulatory system, where everyone chose wildly different things. There's venous blood serum, but all of these things are really basically the same tissue. So we are implementing these SLIMs for all of our different terms so that assay type and disease and all of our other useful metadata terms will also be searchable both at the extremely fine grain that the program thinks is the best fit, but also at a much coarser grain that is useful for data discovery.
Interesting. And I think, Amanda, that Jay Yang has his hand raised. Jay, did you have your hand up?
I'm sorry,
yes.
could you say that again?
I think Jay has his hand up.
Oh, yeah.
Yes. Hi. Thank you, Florence and Amanda. It's great work. I'm not in this area at all, but just very interesting hearing about it. I have some questions. Not sure I asked the right way.
Sure.
So from the get-go, the idea of kind of putting this data together, make it sustainable is great. And all of this multi kind of very grand scale kind of projects. What if you have a small data provider, small project, right? And they may not have abundant of resources. They want to contribute to this, but because they don't have that experience in large scale, they may not code their data the same way, the right way. How would you deal with those kind of cases? I'm asking because I'm thinking about more for cybersecurity kind of data, but I'm curious how you deal with that kind of situation.
Yeah.
So sorry, I'm going to moderator privilege just for a second. I'm going to jump in and just ask if Amanda, you can start to wrap up and then stop sharing so Kenton can get set up. Thanks, because we're pushing time. No, this is fantastic. Thank you. So go ahead and I'll please go ahead and address Jay's question, and we'll see if we can get Kenton set up here.
Yeah.
Thanks.
So I will put this in the chat, which is my best answer, which is that all of the documentation for this model is available and is mutable if you want to take the model and change it for your own purposes. So if people wanted to add their data to our portal, we have a full specification, all of that sort of stuff, and another help system, which I'll put in the chat in a minute, that will guide you through doing that. At the moment, we are only funded to do Common Fund program data, but if we heard from other people that they wanted to contribute data, then like that's just the sort of thing that if we got a lot of interest in it, then Common Fund would be interested in having more data. And so my overall point is just that all of this cool technology is really cool, and it's really useful to have the FAIR principles and to build new models that use the FAIR principles. But you can't just expect that if you take data and have people, if you take a model and say, here, use this model, use these ontologies, that it will automatically make your data fair. You really have to work with the humans that are making those decisions, because they're all going to put their own layer of humanness on how they work with those ontologies. And I will stop sharing and I will answer perhaps John's question.
Sure. First, I commend your bravery that this is a really hard problem. I'm curious about how this fits kind of more broadly. Are there other efforts to either build ontologies or take a different approach to the FAIR problem for this collection of data sets?
Not for our collection of data sets, but I know that other institutes in NIH are also wrestling with this issue. The funding thing through NIH was not specific to Common Fund, so a lot of them have this issue.
Thanks.
So thank you. I'm going to switch over. I'm going to introduce. Thank you very much, Amanda. That was very interesting. I know we've got other questions, I'm sure. And I will also just quickly comment as Kenton gets ready to share his screen. There are actually other technologies being used as well that engage the community to develop what's called a FAIR implementation profile. So they get together and actually across can identify the standards they're going to use to help do this, some of that interoperability. So, but all that for another day. So let me introduce Kenton. He is Associate Director for Software at NCSA, the National Center for Supercomputing Applications at UIUC. For the last 12 years, he's helped to establish a team of research software engineers to support the scientific community broadly in software development, data management, and data analytics needs present in current research activities. I will leave the rest and just introduce him. He can say anything he'd like about his projects. Thanks very much. Go ahead, Kenton.
Thank you, Melissa. I'm assuming everybody can see my screen.
Yes.
Okay, good. So what I'm going to talk about today is the EarthCube GeoCODES effort and a little bit about that program to begin with. So I believe many of you probably know the EarthCube program out of NSF. Was started around 2012, which is a good decade ago now. And it was started to basically try to advance the cyber infrastructure and practices within geosciences. This has basically been focusing around the idea of developing cyber infrastructure, data resources, tools, standards, by which to bring together and use data better within the geoscience domain. So over those that decade, roughly 95 efforts have been funded, spanning all these different areas, developing tools and resources. So with anything like this, there's a common set of challenges that always seem to arise. The most common is around sustainability. So we fund these things, and then what happens after the funding ends? What happens to if in the case of a tool? What happens to that tool? Can you still use it? Exposure. Being knowledgeable of what's out there, what's been funded, and what's available for me as a researcher to actually leverage. That's always been a challenge in the NSF world. And what that leads to is a lot of duplication of effort. Things get built over and over again. And so things like that, tracking relevant products and even packaging things so that they are more readily reusable. That's tricky at times, having to install complex environments and things like that in order to use things, having the necessary resources, stuff like that. So these are pretty common challenges with regards to any of these efforts. And EarthCube has definitely seen these with the projects it's funded, 95 different efforts, what happens after each of those ends. The idea is to keep using these things. So I'm going to talk a little bit about the activities that have been done around that space, and this includes both data and tools. And so first, I would just want to kind of highlight the tech team that's part of the current office, the ECO office for EarthCube. We basically support the various activities out there, try to coordinate activities with regards to data and tools, and foster communication and collaboration, and the annual meeting that happens each year. And I just wanted to kind of highlight that these folks come from 4 different institutions with a vast amount of experience in the NSF world of cyber infrastructure, having developed a number of things within DIBS and CSSI and so forth as well. So there's a lot of experience at hand with this. So on the data side, the activity that is called GeoCODES. And so what it basically is this idea, this came from work with within EarthCube of the Council of Data Facilities. This is the body of repositories that hold some of the larger data sets in geoscience, coming together with the drive to basically make their data more findable and usable. And so what came out of that was this idea of adopting schema.org. Schema.org. And what that basically is a means, it was developed by Google and a number of other companies to basically annotate web pages, landing pages, with regards to metadata that's present there. And so the idea within geoscience and specifically the science on schema effort is to leverage that to annotate data sets. And the point of that is so that they're crawlable. That kind of like Google does with web pages, something can come along and discover this data set and its metadata on each page and build a kind of a master index, so that somebody could come through and basically search across a union of different data sets. And so the idea was to leverage this. It's convenient in the sense that it leverages common web technologies such as JSON-LD. So this is something pretty easy for programs and developers to kind of pick up on and leverage. And so the GeoCODES effort worked with the Council of Data Facilities to basically to help them each adopt it so that their repositories landing pages would be populated with this schema.org. And then prototype out a pilot kind of portal, kind of like a science-specific Google for geoscience to kind of crawl those different data sets. So this was started by the previous SO office, and this is an example of what it looked like at that time. When we came in, we were tasked with basically the task of making more production level, kind of taking it to the next step, making more user-friendly. And so we basically, what we did there is we incorporated a professional UI UX designer. And we took at it an angle of basically dealing with another challenge that we had, which I'll touch on more later with regards to tools, and really the sense that data is really only useful if you have the necessary tools along with it. And so we took the approach of taking the next step of GeoCODES as one of integrating data and tools. And so we gave this to the UI UX designer who worked closely with the office and the leadership council over at EarthCube. And basically asked them to come up with a design that really hit on the user experience and user interaction of this portal. And so they basically took the approach of really simplifying some of the initial prototypes. You know, it really had too much stuff, and really making it easy to kind of navigate, not being bombarded with information. And then that single user interface for both data and tools. As well. One of the things that came out of the work with the Leadership Council also was an interest in being able to see related data sets. And so that was an aspect we incorporated as well, so that basically when you do a search for a data set, find something, you could find something that's related to it in some form or another at the moment based on similar metadata. So this is the interface that was developed, and what I'll do is I'll just kind of walk through a few examples of this. You can go to it yourself here if you go to geocodes.earthcube.org. But then you could do a few examples of different things. So one, you could, a researcher comes by and is looking for high resolution LiDAR data for the July 2019 Ridgecrest earthquake. They could do a search for that. What they do is they go to the page and give it a second and it'll pop up with some results. All right. And so what we have here on the left here is a number of facets. It's a number of them collapsed at first to kind of not bombard the user with ways they can navigate the data, but they are here if they want to get into more depth. So you can do searches based on resource types. So there's both data and tools. And I'll quickly mention that the tools are coming from a number of different sources. One of the key efforts out of EarthCube over the years was the resource registry. So that's the main source of where these tools are coming from. But then you can search on keywords, locations, the specific repository it's coming from. So for example, like HydroShare or others down here, and so forth. And on the right, you have your results. And you can see on the bottom of the results, there's little bullets that indicate whether it's data, and some of them, if you scroll down, are tools, and whether or not they have a kind of a connected tool with it. And this is the kind of the key aspect of the tools, so that you can kind of find the tools that are used with that data set. So if you click on one of the results, you can get the, the usual metadata, an abstract about it, a DOI, if it has location information, where it's kind of located at. And if you scroll down, you can see the tools. So these are some of the tools that are available for it. So if you click on one of these, it'll take you to their webpage if it's the carrying the tools where you have to basically download. Sorry, I'm trying to move my bar here so I can get back to my presentation. And if you scroll down even further, you'll see related data sets. And this, again, is based on metadata, kind of overlaps there. You can click on those, get some summaries of the data. And if you actually want to go to the data set, then you can actually click on that and it'll take you into that data set. And again, you can go even further, find related tools and related data sets to that as well. So another example, in this case, finding a temperature record for pollen and lake sediments from southern Norway. In this case, what I want to highlight is, again, in these kind of connected tools, when they do pop up, some of them are web tools. So in this case, there's one of those, it shows here as a web application. And if you click on that, it'll actually take you to the web tool and if possible, populate it with the data that you were just looking at it. So in this case, it has that information there. There's some errors here, but basically the idea is that you could populate these kind of tools right from the data set that you've been searching for. So if you want to try it, feel free. You could just go here and try it out. This is the current version. We keep it up to date as we add new features to it. On the tools side, we are looking at other ways of basically getting tools. One of the ways we're looking at is basically leveraging one of the EarthCube efforts that was funded called Throughput. What it actually does is it crawls through GitHub repositories and it looks through the documentation present there to try to find links to data sets. And so we use that as another means of kind of finding related tools to data sets. Other things we've been working on in terms of capabilities is the ability to kind of scroll through time. So adding like a scroll bar here with regards to time, in this case, publication year, but also potentially the time with regards to the data set. Additionally, the addition of geospatial capabilities of searching. So maybe a map where you kind of draw boxes and find data sets within that specific area, which would be useful for geoscience research. And then the addition of notebooks. So basically, when you click on a different results, some of them you could pop up with this option of a notebook down here, where if you click on that, it would open up a notebook that has that data set loaded into it, and you can start doing stuff with it, visualizations, analysis, and so forth. And so that kind of takes us into the next part of this. I'm going to skip this for lack of time, but with regards to the actual tools. And so one of the challenges we've had here also is with regards to actually identifying those tools and making them more useful. And so again, with regards to the data set search, that's a useful thing too, in terms of being able to use data. And so over the years, with regards to tools, there have been a number of fronts with regards to how to do that. We had this tools inventory initiative at one point and the current resource registry. And what the effort there is basically to kind of find these tools, get metadata around them, and have them registered into this repository. It's been challenging over the years because it is work. There's a lot of metadata that is possible to fill in with regards to that. And with regards to the people developing these tools, I think it's seen as low value in terms of the effort versus impact kind of aspect of things. And so it's always been a kind of hard endeavor. The office has always had a big and involved in terms of reaching out and helping people basically annotate their resources and get them into the resource registry. And so this has been an ongoing challenge in terms of what do we do about this. And so what came out of this was that one of the kickoff meetings for the new office between the Leadership Council and the Technical Architecture Committee within EarthCube was a number of thoughts. One is we had to do something about addressing the resource registry and getting more tools registered there and making it more user-friendly. There was also this idea of a workbench that was being thrown around by the previous office. And what that was is basically a kind of different kind of resource registry where tools were also usable right from the webpage and maybe connectable in terms of making workflows of tools and so forth. And so that was one thing that was being bounced around as a direction to possibly go in, but there's complications with regards to that. There's also this big push for notebooks that's happening throughout science. And so the idea too was, could we do something there to kind of really leverage notebooks, Jupyter, R, Markdown, you name it. And so in addition to that, there's also this growing push for software as a scholarly object. So it is in itself, scientific output. It's something worth citing. And so there's a growing movement around that. And then from the NSF side, there's always this push for sustainability. We need to keep these activities, whatever they are, going long term. And there's this drawback with regards to the central registry kind of aspect, is that somebody needs to take care of it. And so who is that? How do you fund that? And how do you keep it going? And so one of the interesting notions that came out from a colleague of mine, Clara Narstead at the University of Illinois, was with regards to getting a community to do anything, it's important to really incentivize the community in that regards. And in regards to something like this, the aspect of something that could be on their CV, something worth citing. And what really came out of that was this idea of, well, could we capture tools as notebooks if they're peer reviewed, and by doing the peer review aspect, incentivize the community to do it for us versus us doing it for them. If we had to build all the notebooks for all the tools out there, kind of documenting them and capturing how they run, that would be an incredible amount of work. But if the community did it and they were incentivized to do so, that would be something that would be interesting and potentially sustainable, as the community could keep doing it long term. And so what we did was we issued a call for notebooks as part of our annual meeting in 2020. And so this is the call here, and it was a bit of an experiment. We didn't really know if this would work or not, or if anybody would be interested at all. And so me, Dave Volker, Lynn Schreiber, and Dan Katz, who's one of the founders of JOS, the Journal of Open Source Software, worked on this and issued this call. There was pleasantly some interest from the community. There was some Twitter activity around it. Some people thought it was an April Fool's joke because we made the mistake of making the due date April 1st, but we later moved it to April 15th, but it was no joke. And so at the end of the day, we were pleasantly surprised that we had 21 submissions of notebooks to this call for notebooks. And so we identified a dozen or so reviewers from both the geoscience and cyber infrastructure communities, and we try to do 2 to 3 reviews per notebook. And we ended up accepting 12 of them, 5 of them as oral presentations that were presented at that annual meeting. And we ended up publishing them in collaboration with AGU, the American Geophysical Union, through their eSource system, where they minted the DOIs. So we were pleasantly surprised that there did seem to be interest in it, and we ended up getting a number of notebooks out of it that documented tools that were being developed by the community. There were a number of insights with regards to this. One was runnability. During the review process, a number of viewers had trouble running some of them, so we wanted to address that if we ever did this again. And there was this clear focus on science aspects of stuff versus the tools aspect of stuff. Some were showcasing tools, some were showcasing the science questions that just happen to use a tool, and so you'd see different science questions being answered with the same tool. So we felt the need to kind of address that. And then formatting, with a conference, you expect a kind of consistent look and feel, and we noticed that the submissions were kind of all over the place in terms of look and feel. So having a template seemed to be something we wanted to do next time. We were also surprised that even though we made no restrictions on the type of notebook, all 21 were Jupyter. And so that was something we did in the next call for notebooks that we did in 2021, where basically we thought in terms of runnability, we can then enforce something like the use of Binder, which would encapsulate the execution environment as well. And then by restricting to Jupyter, make it easier for the reviewers to kind of review these things. So we tried that. And then we ended up having 2 tracks too, one for research education and one for demonstration of tools as well. So we did that and expanded the organizing committee and the reviewers because we ended up having way more submissions. So we ended up having 44 abstracts submitted, which is kind of like an intent to submit a notebook. We only had 33 final submissions, which is still more than last year. We think that maybe there wasn't enough time between the abstract submissions and the final submissions, and so some dropped out. But we had a lot more from students and early career folks, which is a good thing. This is what you would expect with any kind of conference kind of proceedings. And we ended up accepting 19 this time, 11 as oral presentations. And so the experiment kind of really worked well. We ended up publishing it this time with a Jupyter book, which is a new technology that Jupyter is pushing out. It kind of makes a group of Jupyter notebooks look like a proceedings, which is convenient for us. And then using GitHub and Zenodo to kind of store the notebooks and minting them DOIs. Another couple of insights came out of this as well, though. One is an emphasis on testing. We had quite a few notebooks that worked when they were submitted, but didn't work when they were being reviewed. And we worked with the submitters to kind of address those if we could. But it raised some issues, that are common with software development, is the importance of testing and freezing things. So that's something interesting if publications go down that route of software. But then again, also we got some feedback from the community that they would like not just to be restricted to Jupyter, which was interesting because the previous year was all Jupyter. But so the idea to support, again, R Markdown as a possibility, even MATLAB for submissions in the future. So that's something we've been actively trying to embrace for the next year's call for notebooks. And so kind of the next steps with regards to this is, community involvement. EarthCube and the EarthCube office is obviously ending. And so the idea is the community really needs to take this up. And so working with organizations such as HEU and this next call for notebooks, we're looking for the organizing chair to be somebody from the community, in fact, not just the office, to really drive this and take this over. And so what basically, I'll close here, but basically what's happened is those tools go into basically our resource registry. It helps us populate in more tools that then get associated with data sets in geocodes. And so we're really trying to push that angle of tools and data kind of going together and really coming from these community resources, data repositories called by schema.org, or notebooks that are provided by the community. And so what has come out of this too is kind of a best practices document put together by the EarthCube Leadership Council, TAC, and Office that kind of outlines these practices over these principles over, I guess, over any specific way of doing things project-wise. Is basically the adoption of standards like schema.org, adoption of notebooks and binder and things like that, just to make things more usable and more fair. So I'll close there. Take any questions that there might be.
Terrific. Thank you, Kenton. Questions? So I just have one quick question, and then we'll wrap up for today. I know there's a couple of announcements, and I need to check something with Chris Daly before we close. So Kenton, I know that you've been working with GitHub and Zenodo to make sure that there's a collection of these that will belong, in a regular repository. Do you anticipate that will continue, or as you're working with AGU, are you thinking, would AGU pick up the collection and manage that, or will Zenodo continue to be sort of the repository, GitHub or Zenodo?
I don't see it as having to be one or the other. We are actively working with AGU, and they are actively working on supporting this idea of notebooks as scholarly objects. So they are definitely going forward with that direction as well. But I see, leveraging things like GitHub and Zenodo and Jupyter Book is another way, especially for smaller organizations, to do this also. So I don't think it's one or the other.
That's great.
Melissa, sorry, can I interject real quick?
Hi, Christine.
Hi. My husband said he'd be quiet a second so I could talk. But going back to Jay's question, Kenton, I'm wondering if you could just say a little bit about how the tech team has worked with the different data facilities, large and small, to try and help them, implement some standards, and because that might be an interesting model for the stuff that he's working on.
Yeah, that rang a bell from the previous presentation. Is that's a big part of what the tech team and the eco office does, and I imagine the previous SO office as well, is with each of the repositories in the CDF, there has been a lot of interaction with regards to supporting the schema.org, but also data cleaning, making it so that things just kind of work together. And so that's an important part of this kind of activity.
Great, thank you. Any final questions? I know we're right at time, and we do have about 1.5 minutes of business just to wrap up. Any final questions? Okay, well, thanks again to both of you for great presentations today. Really important work going on that we all need to know about across the hubs and our related colleagues and projects and organizations. So thank